Okay, welcome back. We are continuing our discussion today on the nucleic acids. And just to remind you where we've been discussing the structures of the heterocyclic bases that form the base pairs in DNA. We've got the purines, which have this 5- and 6-membered ring pattern, with both of them have the ring nitrogens arranged in the same way. But the differences are going to be what hydrogen bond acceptors and donors there are. I will mention that from time to time I get confused because my natural way of thinking about things is nucleophiles, electrophiles, Lewis bases, Lewis acids, electron pair donors, electron pair acceptors, but the language with hydrogen bonding is a little bit backwards. In hydrogen bonding, we talk about hydrogen bond donors, which is the thing that has the electron poor hydrogen that can be donated. But a hydrogen bond donor is an electrophilic hydrogen or an electron pair acceptor. So that's the source of confusion. A hydrogen bond acceptor is a lone pair, which is a nucleophile, which is an electron pair donor. So there's some unfortunate overlap and contradiction in terms of the language, and I will occasionally get that wrong. So just pay attention to the actual groups, and it will be clear. You'll notice how adenine has in red a hydrogen bond donor and then 2 hydrogen bond acceptors, though only this one is important for Watson-Crick hydrogen bonding. Notice that guanine has a hydrogen bond acceptor in blue and then 2 hydrogen bond donors. And all 3 of these are involved in Watson-Crick hydrogen bonding, which we'll illustrate in just a little bit. Then you have the pyrimidines, which are just 6-membered rings. Thymine is in DNA. Uracil is like thymine but lacks the methyl group. And then thymine has this hydrogen bond acceptor and this donor. These are the 2 that are involved in Watson-Crick hydrogen bonding. And then cytosine has a hydrogen bond donor and 2 hydrogen bond acceptors. And the thing we were just getting to at the end of class last time was how thymine has an acceptor and a donor that are complementary to adenine's donor and acceptor respectively. And then cytosine has an acceptor, a hydrogen bond donor and 2 acceptors that are complementary to guanine's acceptor and 2 donors respectively. All right, so that's each of the bases. Now let's show how they hydrogen bond with each other in a DNA duplex. We still haven't talked about what the sugar and the phosphate backbone look like, but that's okay. So what we're going to draw here is a CG pair. So cytosine, the abbreviation is C. All right, something's funny here, so we're going to have to delete and start again. Nope. All right, that's better. All right, so we're going to draw a CG pair. On the left, we'll draw C, and we're going to draw it in a sort of unusual way, sort of tipped on its side. Here is going to be the glycosidic linkage with the sugar. Then here is the oxygen, here is the nitrogen. And here is the exocyclic amino group. All right, so that's cytosine, and I'm missing a double bond there. But as before, we have our 2 hydrogen bond acceptors and our hydrogen bond donor. And then we're going to show hydrogen bonds like this, and the hydrogen bond acceptor, the exocyclic amino group on cytosine, is going to be hydrogen bound to the carbonyl oxygen on guanine. The ring nitrogen that's an acceptor on cytosine is going to be hydrogen bound to an NH bond on guanine, and then same thing here. So we've got to fill in the details, and this is where the drawing gets a little bit uncomfortable. So I'm going to actually draw it all together and try to get everything in the right place, which it will be frustrating, but maybe doable. All right. And I kind of sort of wanted to show the sigma bonds a little more clearly, so maybe we'll just make a little bit more space for that. All right, and then the rest of guanine is here. And here is the glycosidic linkage with the other sugar. Okay, so you have these pairs of hydrogen-bound groups, and this is called Watson-Crick hydrogen bonding. And that's after scientists Watson and Crick, who published the first crystal structure, though it was Rosalind Franklin who actually had who actually did the crystallography, saw the characteristic pattern of sort of this weird X shape in the crystallographic data that they together later interpreted as being the double helix. She wasn't on the 1953 Nature paper, and that's kind of frustrating in retrospect. She also wasn't on the Nobel Prize, but she should have been. And just another example of men of privilege taking credit for the work of others. So that's not, there are still examples of how that's the world we live in, but it shouldn't be that way. Anyway, in the DNA double helix, there's about a distance of maybe 2.8 to 3 angstroms in between the heteroatoms involved in these hydrogen bonds. And that's actually a little bit lower than the van der Waals radii of each of these atoms, the oxygen, the hydrogen, and the nitrogen, respectively, and that's sort of how you can tell it's a hydrogen bond. And then there's about, from sugar over here to sugar over here, there is about 10.6 angstroms from here to here. Now, I forget, I didn't label my notes with this, but one side here is the major groove and the other side is the minor groove. I'm guessing that this is the major groove here and this is the minor groove up here. And there are different functional groups displayed in the major versus minor grooves. So you could imagine that a transcription factor that bound in the major groove might be able to see the other lone pair on this oxygen, the other amino group hydrogens here, and the lone pair on this ring nitrogen, whereas transcription factors that bound in the minor groove might see this nitrogen, the other lone pair on this oxygen, and then the other hydrogens on this nitrogen. So you'll see later on in biochemistry that there are transcription factors that recognize specific sequences, specific base pair sequences of DNA, and in many cases they're doing that as side chains on the protein of the transcription factor, hydrogen bond with groups in the minor and major grooves, respectively. All right, yeah?
Where does the third hydrogen come from on the H2?
Oh, sorry, where does the third hydrogen come from? I apologize. I'm getting ahead of myself a little bit. So I think I was thinking about positively charged ammonium groups in DNA. These exocyclic amino groups are not particularly basic because their lone pairs are tied up in resonance with the ring. So I was just flat out wrong there. Thank you for that correction. Sorry. What else? Okay, so that's a GC pair, 3 hydrogen bonds. We can go through and do an AT pair. Presumably, you could also draw one with a G on this side and a C on this side, and it would be just sort of the same. So what else? Let's do an AT pair just for fun. Because I've got it in my notes, we'll draw the pyrimidine on the left and the adenine on the right, though you could draw a base pair the other way as well. So here is thymine, and in thymine a methyl group is present, whereas in uracil it isn't. So here's the methyl group that protrudes. Again, here is the nitrogen of the glycosidic linkage. Thymine has a carbonyl oxygen here that actually isn't going to be hydrogen bound to anything. And then you have, an NH bond here, and then another carbonyl oxygen here that participate in the hydrogen bonding. So the ones that are going to be involved in Watson-Crick hydrogen bonding are right here. Sorry, I'm the wrong color of red. And then on the other side, you've got the adenine NH group, the exocyclic amino group of adenine. Then you have the ring nitrogen. And then that's all in terms of adenine for the hydrogen bonding. There's just a hydrogen here that really doesn't participate in, that can't participate in any hydrogen bonding because it's just a CH bond, so we're not going to talk about that. Here's the sugar, the glycosidic linkage here. And so again, you've got complementary hydrogen bonds between donors and acceptors, but in the AT pair, there's only 2. And the geometry is basically going to be the same. There's still sort of 2.8 to 3 angstroms between heteroatoms, which is consistent with hydrogen bond formation. And again, there's about 10.6 angstroms between sugar groups or between glycosidic linkages. Again, here is probably, let's see, I forget which is major versus minor groove again, and you can look this up, and it's not that important, and I won't ask you a question about it, but you can see that one of the faces of this base pair for the AT pair has a ring nitrogen and then a methyl group. And these methyl groups can be important for, again, transcription factor and recognition. Apple Pencil battery is low. That's fun. We'll just see how long we can make it stretch. I figure we got 20 minutes before we have to take a break and stand around for 30 seconds while this charges. This is an old edition. Some of you have one of these that you just stick on your iPad and then it charges, but I am not so efficient, so maybe it's time to upgrade. Okay. So those are the AT pairs. Interestingly, if you look at them, and again, I'll just sort of highlight the spacing, 2.8 to 3 angstroms and around 10.6 angstroms between sugars. The AT and the GC pairs are what we're going to call isosteric. That means they take up approximately the same space. That's really important for the DNA double helix because we want the double helix to adopt the same structure no matter what base pairs we have lined up. So it's important that these two types of pairs and then the corresponding, what we haven't drawn is a GC pair and an AT pair, but they're going to be the same as the CG pairs and the TA pairs. It's important that those all be sort of isosteric. They fill the same amount of space. They're sort of interchangeable cassettes in the structure of DNA. Otherwise, DNA wouldn't be a great information storage system if the primary sequence of DNA affected the overall secondary structure of DNA, right? So the secondary structure of DNA is the double helix, and that overall structure stays the same to a first approximation no matter what the sequence of bases is. Though one strand of DNA is going to be complementary to the other. All right, questions you want to ask about that? Yeah.
What about uracil?
Uracil would look just like a TA pair, only the methyl group would be gone. And uracil is in RNA, so RNA isn't methylated here. That does make a difference. I can't remember what it is, and I wish I could tell you. Go ahead.
I think I missed it, but what does isosteric mean?
Isosteric means, steric is related to size. So isosteric means same overall shape and size. So we haven't talked about this yet, but the sugar phosphate backbone of the double helix is here and here. And you've seen the DNA double helix before, though I haven't pasted an image of it here. Probably should have. But it's important for that long-range double helix structure to be maintained that the CG and AT pairs and so on are the same size because they'll fit in that sugar backbone and stack on top of each other. Okay, what else? The questions, go ahead.
Would it also work to kind of flip thymine so the other oxygen bonds with the nitrogen on the end?
That's a good question. Can we flip the thymine so that this one would be here and vice versa? It looks like it would work. There are some issues, I think, with the size of this oxygen and this hydrogen. It's possible that having this methyl group in the wrong place introduces some steric clashes. Again, I don't have a great example of that, and I should know that answer, but I don't. It's also important for the methyl group to be in the right place, whether it's major versus minor groove. So this is the orientation that's preferred. What else? Yep.
Because they're just hydrogen bonds,
Can
can there be mismatches that occur?
there be mismatches that occur because it's just hydrogen bonding? Yes. So there's other modes of hydrogen bonding. What I've shown you here is a Watson-Crick hydrogen bonding scheme. There is another hydrogen bonding scheme called Hoogsteen hydrogen bonding, which I can draw for you. It's in my notes. My guess is you don't care that much, but alternative hydrogen bonding is involved in the so-called Wobble base pair recognition at the third position of a codon, and that has something to do with the degeneracy of the genetic code. We can talk about that more if you want. It's not in the chapter, won't be on the test. Why not? It's fun to take up time in lecture with things that aren't going to be on the test, right? Because then you can relax. Okay, so if you take, we're going to copy this T base, except we're going to erase the methyl group, so it's going to be uracil. And then we're going to see that this U can hydrogen bond in a slightly different way with an alternative partner. So if we rotate the sugar phosphate backbone this way, no, not, yeah, that's how we want it. In fact, That's abominable. Let's go ahead and delete it. They don't like the nitrogens on the side. So keep in mind that this is a pair between, this is a base pair between mRNA, a coding strand, and, no, this is between a coding strand of mRNA and then a tRNA in the ribosome. And under such situations, you can have a uracil. And so the geometry here is going to be different between sugar phosphate backbones, and it's not exactly going to be a double helix. We could look at a crystal structure of this if we wanted to, but we won't. So notice up here, it's the top carbonyl and the nitrogen that are involved in hydrogen bonding. And maybe this gets to your question earlier about whether the other one can be involved. So if it's these two that are involved, we can actually hydrogen bond with the oxygen from guanine and the NH group of guanine. And then this other exocyclic amino group won't be involved. I'm not sure what the quotes are for, Wobble base pair. So U on one strand can recognize G on another strand, or vice versa. And there are sort of 2 hydrogen bonds here instead of 3. Now, what this means, for example, is if on one strand of RNA from 5' to 3', and we'll talk about what that means, if you had the sequence U, 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 the complementary sequence that would recognize that from 5' to 3', we haven't talked about how the structure of DNA is anti-parallel yet. You'll get this in biochemistry. The sequence that recognizes that would be A, right? But what we are saying is that you could also recognize this sequence with AAG. There's some flexibility at the 3rd position in the codon that allows this sequence to be recognized by AAA and AAG. And this turns up in some ways in the genetic code. So if we look at what UUU codes for in the genetic code, it codes for phenylalanine, right? And so the tRNA that binds, the tRNA that has the AAA sequence is the one that carries the phenylalanine amino acid. But if you look at the genetic code, also another thing that codes for phenylalanine is UUC, which means that the AAG tRNA also carries phenylalanine. And so there's the possibility that the AAG tRNA also recognizes the UUU sequence on mRNA, because in the ribosome, the 3rd position in the codon is flexible. There's a little bit of wobble so that you can adopt an alternative hydrogen bonding scheme. So the genetic code is built in with redundancy, and some of that allows for the same tRNA to recognize 2 different coding sequences, and that can be useful for whatever reason. Okay? I probably explained that wrong. There are people who understand it better. They are biochemists. You should go talk to them, but you won't need to for the test because what I just told you about Wobble base pairing is not on the test. Good? You guys like long answers to questions that are relevant but not for the test, right? Decreases your exposure on the test. Okay, well, back to important stuff. Everything I say is important, but anyway, there's a difference between a nucleobase, a nucleoside, and a nucleotide. The nucleobase is what we've just talked about: adenine, guanine, cytosine, and thymine. Okay? The nucleosides are the linkage of the sugar, which is ribose, plus the base, and that's a glycosidic linkage. So it's a glycoside, but because it involves a nucleobase, we're going to call it a nucleoside, okay? And that's just going to be, then we take the ribose sugar and then we put the nucleobase on it, and we're just going to say adenine. And then the ribose sugar is here, and the positions of the sugar are given numbers with apostrophes on them to distinguish them between the numbering scheme of the ring, which you won't actually learn, but there is one. So this is the ribose sugar. The base is linked to the ribose sugar via a beta glycosidic linkage, meaning the base is up. So this part is A. This is the ribose sugar. Notice that the ribose sugar has OH groups on the 2' carbon and on the 3' carbon. And then there is a 5' methylene group with an OH group there. So this is often called the 5' hydroxyl, and this is called the 3' hydroxyl. And in mRNA, the 5' hydroxyl of one nucleoside is going to be attached to the 3' hydroxyl of another nucleoside via a phosphodиэстер. So up here when we were talking about 3' to 5' and so on, that was, we'll show you what that structure is, but it involves the OH groups on the nucleoside. Okay, so when we're attached to the sugar, it's a glycoside, but because you have this nucleobase, which I guess maybe we'll highlight in orange. You've got the nucleobase here, and then you have the ribose portion here. All right, so if it's ribose, this is just a regular nucleoside. If it's got adenine, you're going to combine adenine plus ribose, and you're going to call it adenosine. And we're going to call this a nucleoside. All right. And similarly, if it were C, we would call it cytidine. If it were T, we would call it thymidine. We name the nucleosides differently for pyrimidines than for purines, which is bizarre. And I'm sorry about that. I didn't make it up. Notice that the name of the C nucleobase is cytosine, which makes it sound like it ought to be a nucleoside, but it's not. That's the name of the base. I promise you I didn't make this up, but this is what people talk about. Cytidine is the nucleoside. Thymidine is the nucleoside. These are the pyrimidine nucleosides. And then A, as I've said, is adenosine. That's when it's bonded to ribose. And then G is guanosine. So a little bit weird. The pyrimidines are named. The pyrimidine nucleosides are named differently from the purine nucleosides, and that's just a bit of nomenclature. Okay, if we're in DNA, you've got the same basic structure, only the 2' hydroxyl is gone, and that's where the D in DNA comes from. That's D is for deoxy. So I'm going to draw the same adenine nucleobase, but this sugar is now 2-deoxy ribose, 2-deoxyribose. Okay, and the absence of that OH group actually plays a major role in the stability of DNA versus RNA. It turns out RNA depolymerizes or falls apart into its components pretty easy, because the 2' hydroxyl group can actually act as an internal nucleophile to attack the adjacent phosphoester. And so any of you that work in labs with RNA, you have to be careful with RNA. It's a little bit less stable. The backbone is not as rigid. And actually, there are tons of ribonucleases that chew up RNA all over the place on your fingers. Just because humans are around, we sneeze, and then suddenly there's ribonuclease everywhere. So if you work with RNA, you have to be really, really, really careful. And labs that work with RNA will have a separate set of pipettes, a separate area where you do RNA stuff. They have to use water to which you add an RNase inhibitor, because otherwise even tap water has RNAs in it, and that will chew apart your RNA in your experiment really quickly. So, and that all has to do with the presence of this 2' hydroxyl group in RNA. So the nomenclature for the deoxynucleosides is just deoxy, adenosine, and so on. You just add the deoxy in front of it. Okay, so we'd abbreviate these as DA, DT, DC, and DG. The lowercase d indicates that we lack the 2' OH group. Other questions? Okay, I mean, this is kind of boring. I'm just showing you the atoms for things you've heard about before, but I feel like it's important if you're going to have gone through OChem to know what the atoms in these things are. So what are the nucleotides? The nucleotides are where we add phosphates. Okay, so let's just take, and I'm going to copy and paste to save us time. I know you don't like that if you're working with paper. Oh well, getting used to that by now. You can check this out in the notes later if you want to draw it. Nucleotides have phosphates at the 5'. So if we instead convert the 5' OH group into a phosphoester, what we have here is, we'll name it according to the nucleoside. So we're going to call this adenosine monophosphate. And usually people will put a 5' in there to indicate that the phosphate group is on the 5' oxygen, 5' hydroxy group. And people will abbreviate this as AMP. Okay? So that's the monophosphate version. Now we're starting to get into molecules that you can recognize, because if you take this and then have a phosphoester, but instead of having it be just with one phosphate group, you have it with a pyrophosphate group. Sorry. Clearing my throat a lot today, don't know why. You now have a phosphoester here, and then this kind of linkage is called a phosphoanhydride, or perhaps a pyrophosphate linkage. And just like regular anhydrides are more reactive than regular esters, phosphoanhydrides are more reactive than phosphoesters. Meaning the bond between these two phosphate groups is not as stable as the bond between the OH group and the first phosphate group. We would call this adenosine diphosphate, or ADP. And then, of course, if we add another one on the end of that, oops, you've got the famous ATP, the so-called energy carrier of biology. But it's not just an energy carrier. You need ATP as an ingredient if you want to make RNA. All right. So once you got a phosphate on there, you call it a nucleotide. And then now here is ATP. All right. Question so far? Notice ATP has 2 phosphoanhydrides, whereas ADP has 1 and AMP has none. Yeah?
Which is the form that shows up in DNA and RNA?
Which is the form that shows up in DNA and RNA? Well, we haven't quite got there yet. Because, yeah, and I'll show you that in just a little bit. So stay tuned. What else? Yeah?
With the anhydride, is this what the OH reacts with? Stabilize RNA.
Sure, if you want to synthesize RNA, you will take the 3' OH group from an existing molecule, and you will have that oxygen attack this phosphorus. I'm sorry, yeah, this phosphorus as a nucleophile, and then you'll break the pyrophosphate bond, and then this group leaves, and that creates the linkage in DNA. So we can show you that in just a second. I do want to say one thing, like you probably have been told before in elementary school or whatever, middle school, high school, that ATP is an energy carrier, and that the energy is released when you break a bond in ATP. All right, you break the phosphoanhydride bond and energy is released. And I suppose that's true, Obi-Wan, from a certain point of view, but you have to include, whenever you break a bond, the formation of a more stable bond, right? Because breaking bonds alone, that's uphill in energy. Bond formation is downhill in energy and releases energy. So the thing that releases energy here is that you replace a less stable bond with a more stable bond. So while we're at it, let's show how that process occurs. And again, it's hard to draw all of these bio-macromolecules, so feel free in your notes to abbreviate as needed. But let's begin by showing how we would start the synthesis of a nucleotide. What I'm going to say for ribose applies for ribonucleotides applies for deoxyribonucleotides. So I'm not going to go back and redraw D-ATP, which would lack this OH group. We're okay there, all right? Hopefully. So let's just show some RNA synthesis. I'm going to draw, let's see, guanosine. And then I'm going to show the attack of the oxygen in guanosine on the next nucleotide. Now, this would be taking place in the context of an enzyme called an RNA polymerase. I'm not showing any of that. I'm just sort of showing the chemistry that's going to happen. So, copying all of this. Whoops. And I'm going to erase stuff that's going to get in the way as we're talking. All right, so we're going to show the formation of a dinucleotide by having the 3' hydroxyl on one nucleoside attack this phosphorus. Now, there is some lingo to describe the differences between the phosphoruses. I think, I'm going to get it wrong, but I think it's alpha, beta, and gamma. It might be the other way around. If you learn that someday, hopefully you can forgive me. But for right now, for our discussion, we'll call this alpha. So as you've learned in terpene chemistry, the pyrophosphate group is a decent leaving group, right? So what we're going to do now is just another example of substitution chemistry. We're going to have a base from the enzyme remove a proton from the 3' hydroxyl group. And I'm going to show this mechanism as though it were SN2, where the oxygen here attacks the phosphorus, and then we break this pyrophosphate linkage, which is unstable, and the leaving group leaves. Okay? So what are things going to look like once we do that? I will say that there is some evidence that instead of being an SN2 mechanism, this proceeds via an addition elimination mechanism, kind of like carbonyl chemistry. That is, there is some evidence for a pentacoordinate intermediate in which this phosphorus is still bonded to those 4 oxygens and to that one. That's as though you turned the SN2 transition state into an intermediate. And so that distinction is not going to matter for us. It's going to be simpler to just draw it this way. You may, however, in the future in biochemistry, see people draw the formation first of this bond, intermediate, and then that bond breaks. So just beware of that possibility. When we do that, and now I'm going to abbreviate. We're going to have the adenine base there, the 3' hydroxyl here. The oxygen from that, oh, it's a G there, isn't it? Sorry about that. The oxygen there is going to be bonded to the phosphorus. And that phosphorus itself is going to be bonded to another oxygen, in fact, the 5' oxygen of the next ribose ring. And then this is the A base. Now, we've replaced the unstable phosphoanhydride bond with a new phosphoester. And here is the old phosphoester that was present in the original ATP molecule. This is the alpha phosphorus group. Then over here we've got the pyrophosphate leaving group. And that's pretty stable on its own, though that can undergo a subsequent hydrolysis reaction to give you two phosphate groups. And that also is energetically favorable. Unfortunately, your biochemistry texts will show this reaction as pyrophosphate going to PI for pyrophosphate going to PI. The little I stands for inorganic, as though we need reminder that phosphorus doesn't have carbon in it, but what are you going to do? So what we have now is a phosphorus, the alpha phosphorus from the ATP is now part of the backbone of DNA, and these two esters together are called a phosphodiester. And altogether, this is called a dinucleotide because we have two nucleosides linked together via this phosphorus. Yes?
So in biology, when they say that like DNA is only synthesized 5' to 3', but not if you were to continue this chain, you could only go up. You couldn't do it from the 3'.
Right, so DNA is synthesized in biology 5' to 3', and the reason for that is, for whatever reason, and I suppose you could say, well, why doesn't nature make 3 pyrophosphates on the 3' OH group? And the answer is, I don't know. But the 3' oxygen is always the nucleophile, and then the ATP with the pyrophosphate group on the 5' hydroxyl is always the electrophile. So that's the directionality. It always goes 5' to 3'. Okay, what else? Of course, in DNA, it would be exactly the same, only you would use deoxynucleosides and nucleotides to make a deoxydinucleotide. Okay, so that's structure. What I want to talk about now, and I think that's sort of what's in the book. What I want to talk about now and next time is some things that are not in the book but are nonetheless really kind of cool. So I'm going to show you the sequence of an oligonucleotide. And fortunately, when you see oligonucleotides in the future, they're not going to, we're not going to draw them out for you. We're just going to write the sequence this way. So here is the 5' end of the oligonucleotide. That would be here. And then this is the 3' end. That would be at the very end, and it would have a free 3' hydroxyl group. And everything in between is going to have this basic pattern, but have different bases. And we're going to say this is DNA, so it's got no 2' hydroxyl group. So next time we're going to write the sequence that's complementary here. I've on purpose put an X here because we're going to show you how the stability of a DNA duplex varies when we change this group right here. All right, see you not Friday, but Monday. Have a good spring day.